Okay, well, I've been told that I forgot to introduce myself the first time around, which has been duly noted. I think being the organizer, I sort of imagine that, obviously I don't understand it. But my name is Roman Cholikov, or Roman Cholakov, actually, in Bulgarian. So that's it basically. I'd like to pass the word to Dr. Markov, who's going to be the chair of the first panel, and for the interesting papers.
Okay, good morning everyone, and welcome to the panel on natural sciences at the CantaBulgarian conference. Before we start, I would like to thank and congratulate the Cambridge University Bulgarian Society for the terrific idea to assemble all the best Bulgarian young scholars and the most active ones, and help them present their interesting work, and also meet each other and get to know each other. I like very much the idea. I actually hope there will be energy and enthusiasm for it to turn into an annual event, because it has a great potential. We have an excellent lineup of speakers, which range from astrophysics to neuroscience and to human genetics and vaccines. Because I know the audience will be even more diverse in terms of your interests, and we ask the speakers to try and be as accessible as possible to curious, educated people from other backgrounds, and hopefully this will provide for an enjoyable morning. So after this brief introduction, just to say that everyone will have about 10 minutes for presentation, followed by about 5 minutes for questions. And now it's my pleasure to introduce the first speaker, Velislava Petrova from Cambridge, who is going to tell us about how human genes may affect the effectiveness of vaccines. Have the floor, Velislava.
Okay, hope you can hear me. So I'm Velislava, both in Bulgarian and English. I first really like to thank you to everyone for being here, and for CantaBulgarian President for organizing it and for letting us be part of it. So I'll introduce to you my field, and because I'm now at the start of my PhD education, so it will be more of a preliminary idea of what I will be doing. But it's an interesting and a very specific area that I would like to introduce to you. And I've tried to make it in a very accessible way so that people from different backgrounds would have easy to understand what it's about. So I know that talking about being part of Cambridge is not really relevant, because I'm sure everyone knows where Cambridge is, but I'm not sure whether people know where Wellcome Trust Scientific Institute is, where I work, which is a research genomics institute that I wanted to introduce first. Seems like our presentation doesn't work exactly as expected, so can you change the slide for me, please?
It should work.
Are you pressing the wrong button?
No.
Yeah,
Okay, you can be chatted. I'll tell you when to suggest. So that's the Wellcome Trust Scientific Institute, which is the biggest genomics institute in Europe and the third biggest in the world. And there's around 4,000 people working there in genetics, and there's different areas that it focuses on, starting from cancer genetics, infectious diseases genetics, developmental disorders, different types of scientific areas, all joined by the fact that they're studied from a genetic perspective. So first I would like to start with an overview of what I'll be talking about, and I'll try to divide the talk in two parts and then bring them together. So in the beginning I'll talk about genomics in general and how our understanding about how can we study our genes, what knowledge can we gain from understanding our genes. And then the second part, about more relevant to what I'm actually doing, is the study of infectious diseases. and how we can treat them or prevent them with vaccines or different types of treatment, and hopefully linking the two together of how genomics can be used in order to inform the study of infectious diseases. So does anyone know what this represents? I guess if anyone's from Cambridge, should probably know. No one? Oh, sure you do. So that's what Charles Darwin kind of sketched in his notebook. That's not him. Okay, that's what Charles Darwin did. We studied in very popular kind of even cartoon version of it that he wrote, I think, and then draw the evolutionary tree that completely changed the idea of evolution, while he was just drawing something, probably not considering how important it was. But I would like you to think of evolution as happening now, although we might not see it as an event that is current and that is happening in the moment, but it is actually happening down when you go down to our genes and think about what is happening in our bodies while we are here speaking about it. And if you look around, people around you, you'll see that people are different, and it's very obvious that despite belonging to the same species, to the same phylogenetic class, actually they are very different in their behavioral, in their behavior, in their morphological features, in the careers that they've chosen for themselves. So they could vary, and all these diversities actually encoded in what are we passed to from our parents and what is in our genes, in our bodies. And that's what I'll try to explain to you today. So the first part will be the study of human genome and how we can use modern technology to actually try to understand and read what's within our genes and to see how genes are important in health and disease. So what is a genome? Probably some of you know, and I think that people who are outside of science find it hard to understand where exactly are genes in our body. Do we have them in particular organ in our body, or are they everywhere? Do plants have genes? And this concept of genes is always a bit abstract. That's why I tried to exemplify. Can you just give me the pointer at least? So every cell, and we have trillions of cells in our body, and every cell would have, almost without the germ cells, but almost every cell would have the genetic information from both our parents. So we have this encoded as a sequence of letters, if you want to think about it as letters, and there are 4 letters, while A and Y, A and U are interchangeable depending on what type of molecule it is. It doesn't matter in this case, but it's a 4-letter code that encodes all the information that's within our genes. So we can imagine a very simple code, but actually huge complexity on an organism level. So what's in our genes? People try to think of genes in different ways because pronounced in the same way to work with these different things, but genes seem to be involved in different diseases and also in health conditions, but things like caffeine addiction, everything from chronic disease like Alzheimer's to infectious diseases like flu, also can be involved in prevention or susceptibility to other diseases like diabetes. Also, breast cancer risk could be, breast cancer genes can be linked to something that quite unrelated as obesity. So there's a lot of complexity with it on the genetic level that we actually try to understand in order to understand how disease actually happen and how they evolve, how we can try to treat them. So how do we study our genome? If you go back to the analogy with the letters and think about how do we decide to write a dictionary, for example, we have all the letters in the alphabet, whatever alphabet we decide to choose, and we can use different technology to be able to write. In this case, it could be something really basic, to be able to write actually a whole book where each individual word would have a particular meaning. And that's what roughly genes mean for our body to function. So each individual gene would have a particular function, which would be alternative to the meaning in a dictionary. And then by reading through a dictionary, you can make sense of what's happening in your body. So following this analogy, what happens in our genes is we've now learned in the last several decades to actually read what's in our genes, and that's what happens. That's usually can happen now with some sort of laboratories like these, where they have huge sequencing machine. This is taken from the Wellcome Trust Healthcare Institute, where they have whole facilities with all these sequencing machines that can sequence hundreds of human genomes in several days. And then you can have your whole information on something like this, this flash drive that can hold all the information, all the letters from your genome, and you can carry it around if you know how to use it or how to read it. So this reading of the genome, or so-called sequencing, has become possible because of the development of so-called next-generation sequencing technology, which also so happens to be this process started again at Cambridge and at the Healthcare Institute. And this sort of technology has been, all these graphs, you don't have to worry too much about, except for the fact that the idea is that this technology is developing so fast that we are struggling to have enough computational capacity to store the data that is being available. And also, what it shows is there's such a drop in price. As you can imagine, there are more and more companies being involved, so that the price constantly, it's a very competitive market, and the price constantly drops, and we are somewhere at a stage where a full human genome can be sequenced for $1,000. So it's not even cost problem. So as we have this power, people start to think of scale, and they scale everything up and go up. And if we look at what happened in order to achieve the so-called human genome project, when the first genome of a human being was sequenced, and it took them 13 years to complete one genome. This was before the start of this next generation sequencing technology that I just said, which was around 2005. And then they used all the methods, which took them very long time. Different laboratories in different parts of the world were part of it, so that it was an effort from lots of different countries to achieve just one human genome. What happened now, again, initiative in several sequencing centers in the world, only for 3 years they managed to complete 10,000 genomes. And then David Cameron, 2 years ago, launched this new project for 100K, where he wants to sequence 100,000 UK individuals in order to try to understand what in their genes can define their susceptibility to different diseases or chronic conditions, and in that way to remove some of the burden of the NHS trying to treat these patients. So as the technology is increasing in capacity, people really think much more of numbers now, and it's a game of who can sequence more, but the biggest problem is how can we use this information to inform clinical practice and to inform our study of diseases. So we go to the second part of the talk, which is about what I'm mainly interested in, and is to the study of infectious diseases and how genomics can be a very powerful tool to understand. So that's again an interesting quote in the late 60s from General William Stewart, who was at a meeting of WHO in Switzerland. And at this point, because it was the golden era of antibiotics, people thought that infectious diseases are dead, and it's just a dead field. No one really needs to study them and to fund them anymore. So it's a very, very popular quote from him that it's time to close the book on infectious diseases because they won't be an issue worldwide anymore. But that doesn't seem to be the case. He couldn't have been more wrong. So if we take the incidence of different infectious diseases, as you probably everyone would know from common sense, that the main killers from the infectious diseases side are HIV, malaria, tuberculosis, and in different parts of the world they seem to have different incidence, but it's always the case for most of the infectious diseases that Sub-Saharan Africa, Southeast Asia are the regions that are mostly affected by these infectious diseases, and this is due to different reasons that we can go into if you have questions in the end, but it's primarily, some of them due to lack of vaccines, most of the cases, but for these two there aren't any vaccines, and for most of them actually there aren't. So vaccination is important. You can see here as well in terms of death rate from infectious diseases. And you can see this is Sub-Saharan Africa and Southeast Asia, so there's the majority of deaths from infectious disease occur there. And if you look globally and just follow the purple square here, that's diseases for which we have vaccines, but they for some reason don't reach these populations, or they reach them but they're not administered properly, so people still die from diseases for which we have vaccines. And why don't vaccines get to these people? So vaccines have caused lots of controversy, especially in the Western world, where people start trying to learn more about their health and think that they understand it better, and there are different people giving their thoughts on vaccines and whether they could be actually much more of a threat to our health rather than something to help us. So vaccine efficacy has sometimes been is not very successful because of people's perception of vaccines. And this is the current routine vaccination in the UK. So there are 8 different types of vaccines, and this shows just the age at which the vaccines are administered. And as you can see, for people who don't really understand, it looks like we have too many vaccines. It looks like there's a lot for a person in the early life to take as a vaccination challenge. But giving vaccines to the majority of the population is important. Because of in this case, the case with MMR, this vaccine there was a study here. So this shows how many people get the vaccine, and this shows how common the actual infectious disease is. And here, as you keep high levels of people getting the vaccine, it's all fine, very low levels of the disease. But what happened this year is there was this study that was published that actually linked the vaccine to autism, and people started to think that actually it might make their children autistic. And lots of people actually denied having their children vaccinated, which caused such a drop in so-called herd immunity. This is the number of people in a given population that are affected, that are vaccinated. And as you can see, it looks like a slight drop, but actually, in terms of incidence of the disease, it caused a huge increase, and that's in Western countries. So it's not in a country where people don't understand why they need the disease, why they need the vaccine. So are vaccines effective? There are lots of different studies that have been trying to understand the efficacy of the vaccines and what's important in creating a successful vaccine. But most of them are really more controversy in the general public rather than the scientific world. Vaccines do work, especially because of the scrutiny that they have to go through in order to be accepted as a form of treatment. So what can affect the vaccine efficacy? This could be natural pathogens that could undergo evolution and can actually change so the vaccine wouldn't work or it anymore. It could be that, as I showed you, that actually not enough people get the vaccine, so there's still outbreaks within small population that haven't had the vaccine. Could be some storage or logistics, especially in war countries where they can't have the fresh frozen vaccine that they need. Or host genetics, and that's what my project will be about. How this is very kind of new field of people trying to understand whether, if all these factors are provided, do we still have something else as host genetics to affect our disease? And this can be studied by means of these so-called genome-wide association studies. This is taking huge number of people in the range of thousands, studying their genome, reading their genome, if you wish, and then trying to understand what is the changes that have occurred that make one person more susceptible than another. And that's what my future project will be about. We'll have a cohort of people that will be vaccinated with a vaccine that is just part of the national vaccination schedule. And they will be followed several days after the vaccination, and we'll have a molecular analysis of their blood samples, of their immune response to the vaccination, and also genotyping them, so reading their genome to understand what is the relationship between these two, and hopefully trying to inform what are the genetic patterns that define an effective vaccination. And in the end, if we ask the same question, what's in our genes, it seems like that's always in our genes, but actually what it tells us is that all the information that we need is encoded in this sequence of 4 letters, and all the information actually means life. If we just translate this, that's the genetic code of it. If we translate it, it's life. So if we want to think of ourselves as very complicated creatures, actually it goes down to 4 letters. So I really think we should think about it and try to understand these 4 letters better in order to live a better life. Thank you.
Thank you, Velislava, for a very interesting talk. The floor is open for questions. We have time for several questions.
It's just a question about the graph that you showed.
If we can go back
for
to
the
the graph.
decline in herd
Yeah.
immunization. Why is that increase, that spike, related to the decrease, the perceived decrease in vaccination? Because doesn't it come several years afterwards?
Yeah, so there is a lagging phase after. So initially because there's a slight drop. It's something like 5%. So actually there are a small number of people that are not vaccinated, but actually the majority still are. So you need these people who are not vaccinated to come into contact to create this micro community of people who are not vaccinated to start spreading the virus, because otherwise if you spread the virus to people who are vaccinated, they will just kill the transmission. So it needs some time to actually have an effect, but what it shows that just a slight drop in the overall immunity causes a huge effect on the incidence of the disease.
If you have the genome sequence on sort of a memory stick, what can you do with it?
Well, yourself, or what can people do?
Well, what can people do?
So one study, usually it's done in the context of such genome-wide associated study that I briefly touched in the end, which are, we know that particular genes have been found to be associated with particular diseases. So let's say if you have mutation in this particular gene, you have 60% increased chance of having Alzheimer's at an early onset, let's say. So there's a list of genes that have association provided for them, so they can inform you for your own health and the health of your family members.
But can you do something about it, or it's just more of an FYI at this stage?
Well, there are also platforms where you can send your own, let's say, saliva swab, and they can give you a list of genes and your association to it, your own genome. But the biggest problem is that first there need to be more clinical geneticists who would sit with you and explain to you what this sequence means. So you can do something in cases like very obvious associations, as the case with Angelina Jolie, who decided to have mastectomy because of the fact that she knew that has a mutation and there's 80% increased chance. So in these cases, you make an informed decision for yourself whether you want to pursue this further or you want to just not do anything. I think the problem with genetics is because it's not something obvious, it's a mutation, but there's no big wound on your head. So people usually see disease only if there's morphological feature present. So I think there's a lot to be done in terms of counseling and people understanding what genetics means. I don't think that it's been explained to the general audience good enough for people to know that giving this information is important. I think at this stage it's more important for research purposes where people can use this to develop different treatments or different forms of prevention of diseases rather than being used by people in the general public themselves of going and reading their genes. Was there another one?
More questions?
Yeah, there was another quick question. Okay.
Sure. Just introduce yourself.
Sure. So my name is Velislava. And I was just wondering, so this sort of, I'll ask the same question on graph as well. First of all, if this data is sort of exploring Western societies, assuming that the uptake of a vaccine is high, first of all, like what actually causes the uptake not to be 100% if there's like a national scheme for vaccines? And also, and a hypothetical question, it's a very late question as well. If there's like a micro sort of society where everyone's vaccinated, there's no kind of external sort of source of the disease. If everyone's vaccinated, does that mean that the virus is completely killed, and so you cannot actually?
So these are two questions.
Yeah.
The first question is because we don't need 100%. So the herd immunity is what it's more of a measure in infectious diseases. I mean, I don't want to talk about this in front of him, but it's more a measure when in infectious diseases when you don't need this 100% in order to achieve full coverage of the vaccination, because the incidence of actually having those unaffected people who are exposed to the agent within the same time that can spread the disease is very low. First, you have because you can still decide not to follow this vaccination schedule, so some people decide not to. But as a general campaign, you don't need 100%. You need somewhere, I don't know, 95 probably % to be confident that you would have a good coverage. Then the second question, if I just recall what exactly it was, but it's the idea that if you have a micro community, the fact that the people who are protected doesn't mean that the infectious agent is not present first because they're a small community or they're any community, Second, because it could be, depends on the vaccination, it could be caused by zoonosis, so it could have an animal source, so in the animal reservoir it still will be present. So it's just a matter of when someone susceptible comes. Also, there's no vaccine that will be completely effective throughout your whole life. Most of the, as our immunity wanes, most of the fact that the vaccination campaigns have been successful is because as most of the world is actually vaccinated, there's no more susceptible people. So although the virus could affect if there was someone susceptible, the virus is not around anymore because there's not enough people to spread. Doesn't mean the virus is not present, but if it's not in an animal source or if there's no other small community that they have it, it's just not around because there's not enough people to spread it. So in this case, it would mean that's why that's why smallpox was eradicated. That nowadays, if we have smallpox, probably we'll be susceptible, we'll have it. But because our immunity had changed and also we stopped vaccinating against it. With these diseases, because enough people have been vaccinated, so we don't worry about them anymore.
Okay, well, let's thank, oh, yeah, great.
Yeah.
So knowing the DNA sequence doesn't always indicate the level of mRNA expression, and that doesn't always indicate which genes are going to be expressed. So how do you deal with this confounding factor?
So
Just very quick.
yeah, we can talk about this for hours, but so what these genome associated studies might do two main things. One of them is either look at the particular genes, and as you said, they might be completely the same, but the expression level is different. So this is look at only at the gene structure, and another set of association studies are looking at the regulatory regions around the genes and trying to find mutations around in regions that would actually affect expression levels in promoter regions or in non-coding regions. I don't want to go too much because we'll probably get technical, but the idea is that there are studies that look particularly within these regions that define expression levels, or there's also RNA sequencing, which is you look at the products of the genes, and then you look at the transcript levels of these particular genes. You don't really look at the initial sequence, but what is being produced from it, and then you use what the expression level was based on a number of reads that you have.
Okay, please. Let's thank Velislava for the interesting talk. Our next speaker is Vaccination from Warwick, who is going to tell us about the intricate mysteries of the primary processing of image information from the retina of the eye.
So could you just give me a second? I'll try to fix this if it doesn't look that interesting in the slides. And also, could you try and switching off the microphones when I'm speaking?
Hi, my name is Peter, and I'll be talking about a project I did last year at the University of Edinburgh with Mark van Rossum. So the general topic of the project is the retina and the processing that's going on there. So the retina is the nervous tissue in your eyes. So first of all, I'm going to begin just by introducing you gently to the processing, the relevant processing in the retina, and then I'm going to talk about the problem so that I can set the stage initially. So the problem is that there are more of ganglion cells than non-ganglion cells. I'll explain what these are. Then I'll talk briefly about what the general framework that these kind of problems have been approached, and then I'll briefly discuss some of the results I have. So So how does vision begin? So generally you have a light source somewhere, usually that's the sun, and light from the sun gets bounced off from surfaces